Welcome to FDA Grand Rounds. My name is Rakesh Raghuvanshi in the Office of the Chief Scientist here at the FDA. Today's topic is statistical considerations for drug development in rare disease, and it will be presented by Dr. Emily Morris from our Office of Biostatistics in the Center for Drug Evaluation and Research. There's no reported conflicts by today's presenter in conjunction with today's presentation. I will introduce the speaker now. Dr. Emily Morris is a mathematical statistician in the Division of Biometrics in the Office of Biostatistics at FDA Center for Drug Evaluation and Research. She provides statistical support for the Division of Rare Diseases and Medical Genetics, which reviews drugs and biologics intended to treat inborn errors of metabolism. Prior to joining FDA, Dr. Morris received her PhD in biostatistics from the University of Michigan. We're delighted to have you, Dr. Morris. Thank you very much for presenting today, and the floor is yours.
Thank you very much. All right. Good afternoon, everyone. As Rakesh said, my name is Emily Morris, and I'm a statistician in the Office of Biostatistics in the Center for Drug Evaluation and Research. And my team supports the Division of Rare Disease and Medical Genetics. Thus, most of my experience here at the agency has been with rare disease. So today I'll be talking about statistical considerations for drug development in rare disease. I have the usual disclaimer: this presentation reflects my views and should not be construed to represent FDA's views or policies. So for the first half of my talk, I'm going to introduce some general challenges that we frequently encounter in rare disease drug development, and then focus on the role of the statistician and ways that statistics can help us overcome these challenges. The division I support reviews therapeutics to treat rare inborn errors of metabolism, and many of these diseases fit into the context of interest I'll discuss today. So first, let me set the stage with what that setting is and what I most often see in my reviews. So if we consider a very rare disease, disease that has prevalence much smaller than the 200,000 patients in the US, which is the threshold that's used to be considered a rare disease in the regulatory setting. Instead, consider a disease where we could only enroll about 40 to 50 subjects in a trial. So that's much rarer than that overall threshold. The disease is typically slowly progressive, so it typically manifests over many years or maybe even decades. And there's typically uncertainty about the natural history of the disease. We may not have any natural history studies, or if we have some, there may be very few subjects enrolled in the natural history studies. And then there's often a very heterogeneous disease presentation as well. And so even within families, across siblings, sometimes there's different disease presentation where we may have the same genetic mutation. So it can be very hard to categorize patients even into subtypes when there's this heterogeneous disease presentation. And a few examples of diseases that fit this general context are Niemann-Pick disease type C or NPC, Fabry disease, and Hunter syndrome or MPS II. To make this context a bit more concrete, I'll walk through one example of a specific disease. In this case, we'll use Hunter syndrome just to illustrate some examples in my talk. This is also referred to as MPS II. And this is a very rare X-linked lysosomal storage disorder. And based on my simplified understanding of the disease mechanism as a statistician, not as a clinician, the disease causes sugar molecules referred to as GAGs to build up within lysosomes, and this causes an array of different issues such as breathing problems, heart disease, joint abnormalities, seizures, and neurological decline. There are both mild and severe subtypes that you sometimes see patients categorized into, but typically patients actually fall more on a continuum from attenuated to severe, so it can be hard, especially when you're designing a trial or the patients are early in their disease presentation, to categorize them into the mild or severe subtypes. And disease onset typically occurs between 2 and 4 years old, and life expectancy is substantially shortened for the most severe forms of the disease. MPS II affects approximately 1 in 100,000 to 170,000 male births. Females could be affected, but it's much less common since this is an X-linked disorder. And I'll refer back to this example for some illustrations later on. So in my talk today, I'm going to focus primarily on challenges around the design of a pivotal study used to demonstrate evidence of efficacy and safety. Typically, in rare disease, there's only one adequate and well-controlled trial, so it's especially critical that the design of the single study is scientifically sound. In rare disease trials with a very small sample size, they're often at an increased risk of having an inconclusive result at the end of the trial for various reasons that we'll get into in a moment. So statistical expertise can really help guide the design and optimize the likelihood of conclusive answer as to whether the drug is safe and has a meaningful effect or not. And this means, does the product affect how a patient feels, functions, or survives? In addition to that overall goal of just having a conclusive answer on efficacy and safety broadly, we also want to learn some more specific things like we would in a common disease setting. These are things like who benefits, is there a specific subpopulation that benefits more from this product, what specifically is impacted, what types of symptoms, and also what is the optimal dose. So these questions should also be considered in the design of the trial. Now that I've outlined the disease context and overarching goal of designing the pivotal study, I'm going to describe some of the challenges we commonly encounter. The most obvious one is that we have a small number of patients and therefore a limited sample size for any trial we design. Typically, we have a limited ability to increase the sample size. So often if we're already starting with a very small sample size, we don't have much room to decrease that sample size in order to sufficiently characterize the safety of a product. So in a lot of cases, we're going to assume that we are working with this small sample size. We don't have much ability to change it smaller or larger. And this also affects the number of trials that we can conduct. So I already mentioned we typically have one adequate, well-controlled trial, but this also impacts the early phase studies. So we may have less information when we're designing that later phase trial than we would in a common disease setting where there have been more early phase studies conducted. Another challenge is the heterogeneity of the disease. And this raises questions such as how to select a sensitive endpoint and what the target population should be. If we think of the MPS II example, we may have patients that have mobility affected and strength impacted, but there may be other patients that have neurological impacts. So should we limit to just one group? Or do we want to have a broader population enrolled? And this, in conjunction with the challenge number one of an overall small patient population, it can be very hard to address this challenge. Next, we have the challenge of unknown natural history. And this means we may not have a good understanding of how variable symptoms are, whether there's spontaneous resolution of the disease, how quickly the disease progresses, what the underlying mechanism is, or what endpoints impact patients the most. And I'll just say we really appreciate when patients and patient communities share their insight about what endpoints are most impactful. So particularly patient listening sessions are very informative to help us address that last question. So how can statistics help overcome these challenges? Many of them sound like they may be clinical questions. However, statistical expertise, working in collaboration with clinicians and other disciplines, can really help ensure an appropriate study design and analysis choices are made to provide reliable and timely results from these trials. There may be analytic solutions, or we may use simulations to inform trial design elements. For example, clinicians often like to categorize or dichotomize endpoints to aid in interpretation. However, we know this leads to statistical information loss. Showing the impact of such changes to the endpoint definition can help the team designing the trial to choose the most sensitive option. Simulations can also be helpful to reveal which analysis method is most efficient, and also what duration and sample size are likely to ensure adequate power under reasonable assumptions, rather than an overly optimistic effect size that's back calculated from a feasible sample size. So now I'll revisit each of these challenges and describe some strategies to help overcome them. So first, we have the challenge of a small sample size. One approach is to consider design alternatives to the traditional parallel design. So one example of this is a crossover design, where patients are randomized to a treatment sequence and receive both the investigational product and the control. And one clear advantage of this is that we now have data from all patients for both treatments, effectively doubling the sample size of the data for both treatment groups compared to a traditional parallel design. However, I will caveat that the outcomes are correlated and not independent. So it's not exactly the same as doubling the sample size, but it's still a very useful tool in settings where we can't meaningfully increase the sample size. Some questions related to this design can be informed by statistical modeling and simulations, such as how long of a washout period is needed and what length of time is needed to detect a treatment effect. And there are additional considerations for whether this type of design may be appropriate for your context, such as whether functional unblinding is a concern and if it's reasonable for patients to go off and restart treatment again later. Another powerful tool, another powerful design, I should say, is the platform trial design. And this is a design that allows multiple products to be included in one trial with a shared control arm. And products can enter and leave the platform over time. So the key advantage of this is that the control arm is shared across what otherwise would be individual randomized trials. So one example of a successful crossover trial is for the drug Acnirsa for the treatment of NPC. The pivotal study in this program was a randomized, double-blind, placebo-controlled crossover design of 60 subjects, and all patients were randomized to receive either 12 weeks of the test product, followed by 12 weeks of placebo, or 12 weeks of placebo followed by 12 weeks of the test product, with no washout period. The primary endpoint was the modified SARA score, which is a measure of ataxia, and the target population was limited to those with mild neurological symptoms at baseline. The figure on this slide, which is from the publicly available integrated review for this application, presents the results for both treatment periods and both treatment arms. So along the x-axis, we have the mean SARA score, where lower scores represent better outcomes, and along the x-axis, we have study visit. The blue curve represents the patients that were first randomized to the study product and then received placebo after 12 weeks, and the gray curve represents the randomized treatment group that was randomized to placebo first and then received the study product for the last 12 weeks. The solid line is when they are on the study product, and the dashed line is when they are on placebo. And so I think this graph really illustrates the benefits of the crossover design well. If we focus on the first 12 weeks, so that's from baseline to visit 4, we can see that both treatment groups lowered SARA scores or improved on average. The group that was on the study drug does improve more, but the treatment difference isn't necessarily huge, and it may be difficult to interpret that magnitude when they're both improving. However, when we look at the second treatment period, it becomes very clear that the study product is working better than placebo, because now we see that the patients that were first randomized to the study drug and then received placebo in the second period return back to close to their baseline values, whereas the treatment group that now receives the study drug and first received placebo continues to improve and at a larger magnitude than they did on placebo. So if you look at the two treatment periods together, the overall sequence, it's very apparent just from this one figure that the study drug is working better than the placebo. And then the next challenge that I mentioned is that we have a heterogeneous disease. And there are different approaches that we can consider to handle this, depending on the specific ways in which disease presentation is heterogeneous. So one option is that we can incorporate multiple endpoints. And if we have multiple endpoints that apply to all patients, one statistical approach to help increase power is using a global hypothesis test. And I'll note that a global hypothesis test is testing a different hypothesis than the usual test of multiple endpoints in common disease settings, such as co-primary endpoints, where we want to see an effect on both endpoints. Instead, in this case, the alternative hypothesis of a global test is typically that there's an effect on at least one endpoint. So the use of such a test should be justified by the context, may not be suitable in all settings. But we think in rare disease, it can be very powerful when we think an effect on one endpoint is often sufficient to be convincing that the product is efficacious. So if you have multiple endpoints, but they don't apply to all patients, you can consider a different design where different populations have different endpoints. And statistical expertise can then help you find a way to efficiently combine the information across different subpopulations that are using different endpoints into one statistical test. And that's a very efficient way to do that if there are appropriate endpoints for different subpopulations. You may also consider incorporating longitudinal data so we can observe trends over time rather than relying on one fixed time point. And this, again, statistics can be used to help understand how much is gained by including multiple time points, and the effect largely depends on what type of analysis method is used, which again can benefit from statisticians' help. So for this example, I'm going to revisit the MPS2, a product in MPS2 that I introduced earlier. So this figure here just shows the heterogeneity of this disease. This is from a paper published by Edmond et al. In the European Journal of Pediatrics in 2008, and it shows several different symptoms, their corresponding prevalence from the study of 82 patients, and then the median age of onset, as well as the 10th to 90th percentile of the age of onset. And you can see that there's no one symptom that affects all the patients, though there are a few that have high prevalence, such as enlarged liver and spleen and joint stiffness. However, the variability in age of onset may make it challenging to choose an age group to enroll and a length of follow-up that will detect a treatment effect. So with this type of heterogeneity, it may be helpful to choose multiple endpoints and to enroll a wide age range. And these approaches were used in a pivotal study for the approved product Elaprase to treat MPS II. So Elaprase was studied in a randomized, double-blind, placebo-controlled trial with a 53-week treatment duration. There were two primary endpoints: the change from baseline to week 53 and the 6-minute walk test distance walked, and the percent predicted forced vital capacity, which is a measure of lung function. The primary analysis in this study was an O'Brien rank sum global test, and I'll introduce that in the next few slides, what specifically they used for their test. And the trial did enroll a somewhat wide age range, with the final study including subjects from age 5 to 31 years old. And it was also limited to subjects who had some impairment in their lung function, with FVC limited to those with less than 80%, so that endpoint could be sensitive among the patients enrolled. So the global test that was used in this application is just one example of a global test that we think can be very powerful when there's multiple endpoints and a small sample size. So the O'Brien rank sum test it is very intuitive, so I think it's helpful just to see the algorithm here to understand why this might be useful. So in this case, they had FVC and the 6-minute walk test. So the first step is that across treatment groups within each endpoint, all subjects are ranked. So if you have the best FVC and the best 6-minute walk test, you have the two best ranks for those. And then in the second step, you sum across ranks within subjects. So now we have just one rank sum score per subject, and it falls back into the setting where as if we had just one endpoint per subject. So in step 3, then we can test for a difference in that rank sum score between treatment groups. In this case, they use an analysis of covariance with baseline age group and baseline disease score as covariates, though there are different approaches to actually test for a difference in that step 3. But this general algorithm is how the global rank sum test is used. And then there are many other types of global tests that don't necessarily do the ranking like they did in this approach. So the final challenge that I mentioned is unknown or limited natural history. And there are some statistical approaches to help, such as using different designs, like the crossover design that I already introduced. But another major really useful tool is adaptive design. And this allows you to use accrued information to adapt features of the trial to overcome the knowledge gaps that existed at the design phase. So you're actually accruing that information during the trial and then using that to inform your trial design later on. And this leads me into the next half of my talk, which will focus on adaptive design for rare disease. But first, I think I have a challenge question. So you can just think of your answer, no need to put it anywhere, but this is just to address one of the objectives. So the question is, which of the following are challenges encountered in rare disease drug development? And I'll give you a moment to read the answers and think about which one you think is correct. All right, so if you chose E, you're correct. This is more of a listening check, as those were the 3 challenges I just presented so far. So the second half of my talk is going to be on adaptive designs for rare disease trials. There's a newly published draft ICH guidance on adaptive design, referred to as E20, that was published in June of last year. And according to this guidance, an adaptive design is defined as a clinical trial design that allows for prospectively planned modifications to one or more aspects of the trial based on interim analysis of accumulating data from participants in the trial. Adaptive designs may be helpful when there's uncertainty that remains at the design stage, and they may also minimize exposure to an inferior product by stopping the trial early. But these benefits do come at a cost of increased complexity for the planning and execution of the trial, which may jeopardize trial integrity if not implemented carefully. The guidance describes several different types of adaptations, and I will briefly introduce a few now, but this is not an exhaustive list. So if you want information about more types of adaptations, the ICH guidance and there's an FDA guidance on adaptive disease has a bit more description. So the ones I'll introduce briefly today are stopping early or group sequential designs, sample size adaptation, population selection, treatment dose selection, endpoint selection, and treatment duration adaptation. So first, group sequential designs are where you conduct one or more interim analyses to determine whether to stop a trial early or continue enrolling more subjects. This is a very common approach, probably the most commonly used adaptive design, and it allows for stopping early if the drug has a larger effect than expected, or to stop for futility if the trial is unlikely to demonstrate efficacy. And it's been shown in simulations and analytically that it minimizes the expected sample size and minimizes the trial duration as well. So this is a very efficient approach. When it's applicable, it's a very useful tool. So it's helpful primarily when there's uncertainty about the treatment effect size, and it is you can apply it when there are enough patients to increase the maximum sample size. So sometimes in rare disease, if we are already enrolling a very small number of patients and we don't feel like we can really increase it, we don't know that having an interim analysis with a smaller number of patients would be meaningfully improve the ability to characterize the product. So sometimes this can be applied, but in some of these very, very rare diseases, the group sequential design is not as commonly used. And sample size adaptation is similar to group sequential design, where you conduct an interim analysis to determine whether the final sample size should be increased. They're both comparative or non-comparative options, and that means that for like a non-comparative option, you would use pooled information rather than have treatment groups compared at the interim analysis. So you could look at the overall variability of the primary endpoint, and the advantage of the non-comparative approach is that it maybe has less risk to trial integrity as the treatment assignment remains unknown to those conducting the analysis. And the comparative approach means you're actually looking at, treatment A versus treatment B, and then using that analysis to inform whether to increase the sample size or not. And a very common approach for the comparative method is referred to as the promising zone and uses the conditional power approach. This is very helpful when there's uncertainty at this design stage about estimates of treatment effect that impact the sample size calculation, things like the treatment effect size, the variability of the outcome, placebo response rate, etc. But it also requires that there's enough patients to increase the sample size, just like in the group sequential design setting. So conditional power, which I mentioned briefly, is a common approach to this, is defined as the probability of rejecting the null hypothesis at the final analysis, given the test statistic that's observed at the interim analysis. And in this case, we're calculating that, assuming that the treatment effect estimated at the interim analysis is the true underlying effect. There are other definitions of conditional power, but the overall idea is that based on accrued information in the trial, you can update your power calculation and determine whether the trial will be underpowered or sufficiently powered with your set pre-specified sample size at the start of the trial. So conditional power is then used to categorize into promising, favorable, or unfavorable zones. And then based on which zone it falls into, the sample size is increased or remains the same. And you can see in this little figure on the right that the promising zone is sort of that middle zone. And if it's either favorable or unfavorable, the sample size is not increased. And the advantage of this approach is that depending on how these zones are selected and the significance level, you may not need to adjust your alpha spending in order to control the overall type 1 error of the trial. So that can be very appealing. However, there are some critics of this approach because for various reasons, but one reason is that it feels very counterintuitive for the conditional power to fall into the unfavorable zone, meaning your trial is likely to be underpowered for the actual effect size. And so it seems like that should also lead to an increase in sample size, but with this paradigm, it typically does not. And so that is one criticism of this, and one reason group sequential design is often favored over the promising zone approach. But these are both viable options and very useful in certain contexts. And like group sequential design, you can also incorporate stops for efficacy or futility early in the analysis as well. The next type of adaptation is population selection. And this is where you can conduct an interim analysis to determine whether the trial should be limited to a specific subpopulation. This is helpful when there's uncertainty about which patients are likely to benefit from treatment. So if we think about having a broader treatment population and then a certain subpopulation we're pretty confident the product is going to work for, but we're not sure if it's going to work for the broader population. So we have two options. One, we could consider conducting the trial in the broader population, but if the product ends up only being effective in the subpopulation, this may lead to an underpowered study because we're effectively just infusing noise by enrolling all those patients into the study that it will not be effective for. And so on the flip side, if we conduct this trial just in the subpopulation of interest, but we don't test it in the broader population and it ends up being effective in that broader population, this would unnecessarily restrict the indication and access to this product in a population it could potentially help. And so instead, we may consider doing an adaptive design where we enroll all patients from that broader population at first, and then conduct an interim analysis to determine whether we should restrict to the subpopulation of interest for the remainder of the study. The next type of adaptation is treatment or dose selection adaptation. And this may be applied when there's multiple test products or multiple doses of a test product that are still being considered in the pivotal study. So I will note that this should not replace the appropriate dose finding studies to be conducted earlier in the drug development process, but it can be helpful when there's still uncertainty about the benefit risk profile of more than one treatment. Endpoint selection adaptation allows the primary endpoint to be chosen after an interim analysis, which considers multiple possible endpoints. And we note that the rule which determines which endpoint is selected should be fully pre-specified, and statistical testing does need to account for the multiple tests conducted at the interim analysis. And though this is true of all of these adaptive designs, they should all be fully pre-specified with the rules that will make decisions on adaptation very clearly described prior to initiation of the trial. This is particularly emphasized with endpoint selection, as we don't want a trial to be ongoing and then one endpoint that looks promising at the interim analysis is selected for primary. This should all be really fully pre-specified. So this can be helpful when there's uncertainty about the treatment effect size of different endpoints, and there are multiple acceptable primary endpoints that could be used in the study. However, I find oftentimes that just using multiple endpoints for the final analysis as well may be a more appropriate solution rather than requiring an interim analysis to select just one to move forward. So finally, the last type of adaptive design I'll talk about is adaptive treatment duration. And this allows for the length of follow-up to be increased after an interim analysis. So essentially in this approach, at the interim analysis, you conduct it when all subjects have reached some interim length of follow-up. And if the test is statistically persuasive, you can stop the trial early at that duration. But if it's not statistically persuasive, then you continue following patients until they reach longer follow-up. And we think this approach is helpful when there's uncertainty about how quickly the disease progresses with or without treatment, which is often the case as I described in the diseases that we encounter in my reviews. So thinking back on all of those different adaptive designs I just introduced, we think one of the most widely applicable adaptations in the rare disease context is adapting the treatment duration. So we already stated that it may be hard to increase or decrease the sample size to use an approach like group sequential designs or adaptive sample size. And then also population selection may not be feasible if we already have a very small population to begin with. And treatment and dose selection may not be applicable if we don't have multiple treatments or multiple doses of interest. And endpoint selection, again, may not be applicable if we don't have multiple endpoints that we were trying to choose from. So we feel that treatment duration adaptation is pretty widely applicable, and especially useful when we hear hypotheses that the product will have a larger effect size the longer patients are treated with it. So this approach can be helpful in that type of context. So several members of my team and my office have worked on research to evaluate this adaptive duration treatment approach. And we don't have a public example of this being used in a pivotal study, so I'm just going to present simulation results here. But we have conducted research to understand this approach and see where it might be useful. So I'll present that in the next several slides. So the example setting that I'll use for the simulations is described here. We have roughly 40 patients enrolled in the trial. They'll be randomized 1-to-1 to placebo or study drug. We'll assume we have a continuous endpoint, though this can be generalized to other types of endpoints as well. And we'll assume that there's a linearly increasing treatment effect over time, followed by plateau. So the figure on the right kind of illustrates what this would look like. So patients, as soon as they start treatment, they start to have a treatment. There is a treatment effect observed immediately, all the way until 2 years of treatment in this case, and then it reaches its max treatment effect. In this case, I'm plotting the standardized treatment effect so that we could compare across different treatments. So this is basically the treatment difference divided by the variability. But the idea here is that the longer these patients are on treatment up until 2 years, the further apart the two curves for placebo and study drug should be, the clearer that treatment difference becomes over time. So then if we're designing a trial under that setting, we may wonder, how long does the trial need to be? We could consider a 1-year trial or a 2-year trial. And on this figure, I'm plotting the power for just a fixed duration time of 1 year or 2 years. And the orangish yellowish curve is for the 1-year trial, and the reddish curve is for the 2-year trial. And so we can see here that 1-year trial is going to be pretty underpowered for most reasonable treatment effects. That standardized treatment effect of 1 at 2 years, which is what I showed in the last figure, is that center line. So at that point, the power of the trial that's only 1 year long is only 33%, whereas it's 88% if we conducted that 2-year trial. So it's pretty clear from this that we need to conduct a 2-year trial if those assumptions are correct about the treatment effect. However, because we may still have a lot of uncertainty about the treatment effect, we might instead consider doing an adaptive duration approach, where we have a 2-year trial, but we conduct an interim analysis when all patients have 1 year of follow-up. And then if the results are persuasive from 1 year, we could stop the trial early. If not, we continue on to that planned 2-year trial. So one thing to note about this type of design is that our outcomes being tested at the interim analysis and the final analysis are different. So in like a group sequential design setting, you're testing the same endpoint just on a different number of patients. But in this approach, we actually have different endpoints. So though they are different endpoints, they're likely very correlated. And so there is still a multiplicity issue if we conduct 2 tests overall in this trial, and we need to account for that multiplicity so we don't inflate the type 1 error. So our team wondered if we could use some ideas from group sequential design methods to account for the multiplicity in our adaptive duration design. So I'll go back to group sequential designs for a moment and explain some methods here. As a reminder, group sequential design is where you can conduct 1 or more interim analyses after a pre-specified portion of subjects have the final endpoint observed. And this is referred to as the information fraction. So if you could think of a 50% information fraction corresponding to 50% of the subjects have the final endpoint observed, and then we conduct that analysis to determine whether we want to continue to enroll the other 50% of subjects. So as I mentioned, this is different from the adaptive duration, where we actually test the same endpoint at each analysis. So there's definitely a multiplicity issue in this context, and there have been many approaches that are very well established that are known to adjust for multiplicity appropriately. and can be used to control overall type 1 error of the trial. So two of the approaches that we'll use are the O'Brien-Fleming method, which spends a small amount of alpha at the interim analysis, but reserves most of that alpha for the final analysis, and the Pocock threshold, which spends alpha evenly across each analysis. This figure here illustrates how the differences between those two approaches. So on the figure on the right, we have the critical value along the y-axis. That dashed black line represents the critical value if we were not to adjust for multiplicity at all. And then the green and blue lines represent the Pocock and O'Brien-Fleming approaches respectively. So we'll focus on just those two. And then in the x-axis, we have the information rate or information fraction. So we can think of 0.33 as when 33% of patients have the final endpoint, and 1 is when all patients have been enrolled and observed for the specified duration. So on the left, if we look at the green dot, we can see that's closest to that dashed line. So that means it would be easiest to show a persuasive result using that Pocock threshold at the interim analysis. And on the converse, the O'Brien-Fleming dot, the blue dot, is the furthest away. So it requires a very persuasive result to actually stop early using the O'Brien-Fleming thresholds. And then if we go all the way over to the right, where at the final analysis, where we have all patients, now for Pocock, since we've already spent some of our alpha, that one is now the hardest to show a persuasive result or to consider it statistically significant. And the O'Brien-Fleming dot, it's hard to see, but it's very close to the critical value if we were not to adjust for multiplicity at all. So essentially, you're able to test at the final analysis at almost the full alpha level as if you didn't even have the interim analysis previously. So that can be one reason why that one is really appealing, but if you really want to stop early, the Pocock thresholds are a bit easier to show a statistically significant result at an interim analysis. So we were thinking, can we apply similar rules to adjust alpha at the interim analysis for the adaptive duration approach? In this case, our information fraction is slightly different. We were going to use a concept of the length of follow-up as the information fraction rather than the proportion of patients enrolled. And so these two figures here just kind of illustrate what data is being used and how it's a bit different between the two approaches. So on the left, well, in both figures, each horizontal line represents one patient enrolled, and it's their calendar time in the trial. So in this case, I'm assuming an enrollment rate of 2 subjects per month, and we need 40 patients total. So we have in order to enroll all subjects, we need 20 months until the last 2 subjects are finally enrolled. And then we do our interim analysis after 12 months. So we can conduct that interim analysis essentially 32 months after that first patient is enrolled. And the orange represents that we have data from all 40 subjects when we conduct that interim analysis under this design. And then on the right for group sequential design, because we base this off of 50% of subjects, but we need them to have 24 months of follow-up, it's a bit different, but the timing of the interim analysis ends up pretty similar just by chance because of the way that this is set up. So we only need 10 months to enroll all 20 subjects we'll use for the interim analysis, but then they need to be followed for 24 months. So we end up conducting this interim analysis 34 months after the first patient is enrolled. But you can see we're using just half the patients rather than all of the patients, but the duration is longer. So we did apply O'Brien-Fleming and Pocock thresholds to see if we could control the type 1 error under the adaptive duration approach. And this figure here presents the type 1 error for the 2 different designs and the 2 different thresholds. And we can see that at least in this example setting, it does appear to control type 1 error across all different correlations between the 1-year and 2-year endpoints. So we would expect that if the endpoints are more independent, this would be more likely to inflate our type 1 error. And if they're more highly correlated, then it would be a more conservative test to account for multiplicity. So this is encouraging to see. It seems like we can apply the Pocock and O'Brien-Fleming thresholds to adjust our alpha level and still preserve the overall type 1 error of the trial. However, we do caution that simulations are likely needed for the specific context to demonstrate that type 1 error is not inflated. This is just one example, and we have not looked comprehensively at every possible iteration that you might have with different types of endpoints, a different number of endpoints, different sample size, things like that. So though this result is promising, we would still like simulations to justify it for a specific context. And then now that we think type 1 error is well controlled, we are interested in what the power of this type of design would be. So in this figure, I'm showing the power versus the standardized treatment effect at 2 years. Again, that standardized treatment effect of 1 we assumed for our example setting is that middle line. And I'm comparing the adaptive duration approach for the O'Brien-Fleming threshold and the Pocock threshold to that fixed 2-year trial that we looked at in that first figure. So what we can see is that the green and red lines are nearly overlapping, and that is the O'Brien-Fleming used with the adaptive duration approach, and that fixed 2-year trial have almost the same power. And this is really encouraging to us because it means that we can do this adaptive duration approach and still look at an interim analysis at 1 year without too much cost to the overall power of this trial. So, you know, it could be really advantageous to have that chance of stopping early, but we're not necessarily losing overall power from this trial. The Pocock method does have lower power, and this is true in group sequential design as well, I believe, where when you spend that alpha earlier, it affects the overall power of the trial, and O'Brien-Fleming is typically the more powerful approach. So you may wonder, you know, why use the Pocock method instead of O'Brien-Fleming? But it has a much higher chance of stopping early. And so this figure shows the probability of stopping early on the y-axis by treatment effect again, and at that 1 year standardized treatment effect, the power or the probability of stopping early is 25% for the Pocock threshold and only 9% for O'Brien-Fleming. So it is a trade-off between power and probability of stopping the trial early to determine which type of adjustment is appropriate. And then we also were curious, you know, how does the power of this adaptive duration approach compare to group sequential design? If group sequential design is an option, you know, how would we decide between these two approaches? And this figure here presents adaptive duration versus group sequential design for both O'Brien-Fleming and Pocock again. And the dashed line is the group sequential design, the solid line is adaptive duration. We can see that they're very, very similar. The lines are very nearly overlapping, but group sequential design does appear to be consistently slightly better in terms of power. So if that is an option, it's probably better to use the group sequential design. However, in settings where that's not necessarily applicable, this adaptive duration design can be a really useful tool, and we were encouraged to see that the power is quite similar to group sequential design. So the overall takeaways of the simulations related to the operating characteristics are that the adaptive duration with adjustments used from traditional methods in group sequential design appear to have the type 1 error well controlled for this setting. The power for the O'Brien-Fleming adaptive duration design is similar to that of the fixed duration trial at the maximum length, and the power for group sequential design and adaptive duration are similar, though group sequential design may be slightly more powerful. So some additional considerations about using this type of approach. There are different characteristics of the trial that may change these conclusions, such as the correlation between the earlier and later time points being tested at the interim and final analysis, the different analysis methods. If you were incorporating longitudinal data, this could really change how much is gained with this approach, and then also combining features to increase power, such as using multiple endpoints or longitudinal data, can also change both the power and type 1 error. So we would really encourage simulations to investigate that for the specific context of interest. And then also, just before using adaptive duration design, we think that you should consider the ability to conduct that longest fixed duration trial versus adding an interim analysis. And I think the ICH guidance goes into this well about a lot of the risks for doing any sort of adaptation. And if there's not that much gained by conducting that interim analysis, it may be a safer option to conduct that longer fixed duration trial. But if you are going to do an adaptive duration trial, then you also need to weigh the desire between stopping the trial early with the overall trial power, since we see that that is a trade-off depending on what threshold is used. And also the enrollment rate will have an impact on whether the adaptive duration design is appropriate or beneficial. And I'll just very briefly show that, as I know I'm getting close on time, but I'm nearly at the end. So in this slide, I'm showing the difference between an enrollment rate of 2 subjects per month on the left and 1 subject every 2 months on the right. So on the left, the interim analysis occurs when 40% of subjects have complete data, whereas on the right, 85% of subjects already have complete data at the time of the interim analysis. So you can see that it may not be that beneficial to conduct an interim analysis that late in the study, and it may be better to look at something like group sequential design that doesn't require full enrollment of all patients for when you have a very slow enrollment rate like this. So that's just another thing to keep in mind with this type of approach. And some additional considerations for adaptive designs in general from the guidance. And I think the guidance goes into much more detail about this, so I recommend reading that if you're using adaptive designs. But they emphasize that maintaining the trial integrity will be challenging. Just knowledge that an adaptation occurred can sometimes influence behavior and affect your results. So that really needs to be, there need to be guardrails up to prevent that type of impact. You should also ensure that there's a clear role for the adaptations, that the independent data monitoring committee can make the adaptation recommendations, because if it's not understood, that could really affect the trial results at the end if the adaptation does not go as planned. And then also you should ensure that there's sufficient information for full benefit risk analysis. So I primarily focused on efficacy, but the safety is a huge concern as well. And you have to ensure that that interim analysis would allow for the full benefit risk, including that safety analysis. And you also need to be careful, particularly in rare disease setting, that you're not, that the interim analysis is not based off of too little or too early of information, such that the estimates are highly variable and not reliable. Because making an adaptation on something that is unreliable can lead to, an unfortunate selection of subpopulation, or, in a lot of these cases that can be risky too. So you have to be careful that it's not too early and based on too little information. And then finally, the guidance also emphasizes just to limit complexity of adaptations in a confirmatory trial in general. Again, related back to that first point that trial integrity will be more challenging. So if you limit complexity, that makes it less challenging, less factors to consider for the trial integrity. So in conclusion, we encourage conducting simulations to evaluate the operating characteristics of any non-standard design, particularly the adaptive duration design, and also to consider aspects that can be adapted where there's an information gap at the design phase. Because there's often many information gaps at the design phase in rare disease, it's best to choose just one, have one type of adaptation to reduce that complexity of the trial. And there will always be case-by-case considerations. All of the complexity increases if you use multiple endpoints or conduct multiple interim analyses. So again, it's really very helpful to consult with a statistician to get insight onto the specific context of interest. And I have one final challenge question. Again, just think of your answer. You don't need to put it anywhere, but what is a key advantage of the adaptive duration design? A is that it's the most powerful adaptive design approach. B, it does not require sample size to be increased. C, it accommodates uncertainty in how long it takes to observe a drug effect. And D, B, and C. And the answer is D. It is not necessarily the most powerful approach, as I mentioned previously. So I just want to highlight a few guidance documents that I referenced or used in my slides. The first one is ICHE 20 that I talked about. There's also a rare disease guidance, considerations for the development of drugs and biological products that can be useful for those working on rare disease trials. An FDA guidance on master protocols, which goes more into detail about the platform trial design that I briefly mentioned, and an FDA guidance on adaptive design, which has a little bit, which is very consistent with E20, but does have some information that I used in my slides. And then I've also presented here just some stats papers that have methods that I either mentioned or used or can kind of help address some of the challenges I mentioned for rare disease that I didn't talk about in the talk today. And I just owe a special thank you to some colleagues in the Office of Biostats who helped with the research and with preparation for this presentation: Yan Wang, Lainey, Rebecca Chu, Emily Nguyen, and Greg Levin. So thank you to them. Thank you all for listening.